Hello, my name is Sally Ciccini, and I had the opportunity to be mentored under Dr. Guang Ru at UNC Chapel Hill and on an independent research project entitled The Impact of Comorbidities on Brain Aging Trajectory and Alzheimer's Disorder. And essentially, Alzheimer's disease can be characterized by a couple of biomarkers within your brain. Essentially, there is neurodegeneration, so your neurons are not able to send signals to one another and they kind of die off. There's also tau proteins that create these wirings in your brain called oblongers, and here is a neurofibrillary tangle of these tau proteins which cause this brain rewiring. And then there's also some cortical thinning, which are critical parts of your brain that as you get the disorder, they're not able to perform their functions because they are thinning, and we're going to focus mostly on cortical thinning within this research process. My question in this research is, how do the brains of those with Alzheimer's patients with other comorbidities, so if someone has Alzheimer's but they also have a different disorder such as diabetes, hypertension, any cardiac disorder, or even anxiety, how does this additional disorder or several disorders impact the brain aging trajectory in Alzheimer's patients compared to those that are cognitively normal? Additionally, some objectives that I have are identifying the differences between Alzheimer's patients and cognitively normal patients, and then essentially applying that to comorbidities and how identifying specific comorbidities that impact Alzheimer progression. Some of the methods that I used are analyzing the neuroimaging data that I got regarding a lot of the patient's medical history and cortical thickness data. This is provided from the UNC Neuroscience Hospital. And these samples of cognitively normal patients and Alzheimer patients are separated and by gender and whether they are of course, diagnosed with Alzheimer's disorder or not, and then I used RStudio for some statistical analysis. Additionally, some of these results, as you can see, the blue are cognitively normal patients, and here is a graph with only Alzheimer's patients with the red, and we can see under this, if this is the normative modeling line, we can see that the Alzheimer's patient's cortical thickness, the higher the better, is significantly lower than typical patterns, which we can see, and in the future we will look at why are certain patients going way lower than normal. And as you can see here, what's interesting is there are patients that fall way under that have Alzheimer's and way in the middle. So as we can see here, patients experience Alzheimer's in different ways, and no brain is the same even within a certain disease. Additionally, some results here, if the green is cognitively normal, we see that the deviations are way bigger within Alzheimer's disorder patients, having a greater deviation from the normative brain model. Some conclusions that we have that these findings highlight the importance of using a normative model to assess brain aging. Essentially, we can see greater differences between the Alzheimer's patients and then cognitively normal patients. And then we can use neuroimaging data and multimodal modeling data to assess and understand these diseases better. Some implications are normative brain aging models prove useful and they are essential for these comparisons again. And then patients experiencing Alzheimer's disease differently, we can understand exactly how every single patient has a different brain experience, and we can understand that their medical history and their combination of all of their experiences are a way that we can better understand what they're going through and their disease progression. Additionally, some future instructions are a longitudinal study essentially predicting cognitive decline so that people can get better treatment, especially a treatment that is personalized for their needs. And then future studies on specific comorbidities impacting Alzheimer's disorder progression. If we're able to understand how a specific disease might impact Alzheimer's disorders a bit more or less, we'll be able to provide specific treatment. Additionally, we can understand how these disorders might have a link to Alzheimer's. For example, in my studies, I found how specific cardiac disorders have a great impact on Alzheimer's disorder which we can apply to this further research. These are some of my references, and I'd like to thank you for listening.